Okay, so we'll start here with what it says. If an organism has a dominant or the dominant phenotype, how do you know whether it's homozygous dominant, capital B, or heterozygous dominant, capital B, lowercase b? So we need to do a test cross. It is used to determine the genotype of a particular phenotype. In other words, to find the genes behind the physical characteristics of something. Phenotype being the physical characteristics, what it looks like. P here stands for parent. Generation. Okay. So what we're trying to find out is what is this second gene here? We're going to test. It says here by crossing whatever this is. Okay, we have two possibilities. There are two possibilities. So it's either TT, that's the parent, or T lowercase t. We don't know which one. So to determine, this is one genotype, this is another genotype, both of the same phenotype, both which would result in the same physical appearance. So it says here, by crossing mean one of these two possibilities, with a homozygous recessive individual, you can infer the genotype by observing the offspring. So let's take a Punnett square and do that. We're going to cross the big T with the homozygous recessive, which is little t. So let's fill out this Punnett square. We get here big T, little t. And on the bottom we get big T, little t, big T, little t, big T, little t. This is the first cross. They're all tall because they all have at least one big T. Okay. So the big T represents the tall gene and the little t represents the short gene. Since there's at least one big T in all of these pairings, we know that the offspring are all going to be tall. Since they're all tall, we could, again, if we saw this, that all the offspring were tall when we cross whatever the parent was with the little t, we could then infer that the original parent had this combination, big T, big T. Okay, let's now cross. Let's see what would happen if we crossed a parent with big T, little t. I'm going to use this Punnett square. Copy it, see if that works. And then erase everything in here. I'm going to cross the big T, little t with homozygous recessive individual. We'd end up with this. In the upper left-hand corner, big T, little t. Down below, big T, little t. And then here, little t, little t, little t, little t. Just to make this a little more clear. Okay, so this F1 is this generation is 50-50, 50 of each. 50 tall, big T, little t is tall, and then 50 short, little t is short. Right? There's 2 of each, which means 50% of each. It could also represent as a ratio 1 tall to every 1 short. You could write 2 to 2, but that reduces to 1 to 1. 1 tall for every, in simple language, for every tall, you get 1 short. So what can we conjure, what can we infer or confirm if we had this ratio of offspring? 50% tall, 50% short. We could confirm that the parents must be heterozygous. They must be this. Big T, little t. As opposed to if they all ended up tall, we could confirm that the parents must be of the genotype big T, big T. Okay, so we can now go on to what's called dihybrid crosses, which involve 2 genes for 2 traits. So in this case, we're going to look at yellow and green alleles and round and wrinkle. Yellow is dominant, round is dominant. And we're going to imagine a cross between a yellow round, which is homozygous dominant, and a green wrinkled parent, which is homozygous recessive. So it's these 2 here that involves these 2 here. So that's the homozygous dominant and homozygous recessive. And predict what the F1 generation is. F1 generation isn't too hard. It's a mixture of both. So you're going to end up with the following for F1. Yellow, green, round, wrinkled. That's your F1 generation when you cross those two sets of genes. Now we're going to look at what happens when we cross two F1 generations. So when we cross this with itself. YYRR. So what will we end up with? Well, we'll end up with a combination of the alleles. So each parent's going to contribute a YR, right? That and that. And then a Y little r, big Y little r. So I'm doing this. I'm starting off using like a FOIL rule. Big Y, big R. That's the first contribution. And then big Y, little r. It's the second contribution. And what are the other combinations? Well, other combinations are little r, big R. Let's fix this here. And then the last combination is little r, little y, little r. Okay, and that's what we have here. We've already got them placed in the Punnett square, both horizontally and vertically here. And we're going to cross them just to see what we get. So we'll cross them and we'll see. Cross this first one with this one here. We get big Y, big Y, big R, big R. Keep in mind that, let's see here so we're clear. I highlighted it here. Big Y indicates, uppercase Y indicates yellow, and then uppercase R represents round, lowercase y represents green, lowercase r represents wrinkled. Okay, so let's go back to this cross here. We'll cross this one here. And then this one here. And then this one here. Okay, and do the same thing, start again here. And then I'll work my way down, starting with this here. So we get and then here. little r, and then here, and then here. I made a mistake here. Little r, little r. And then here we'll start with the little r or little y, big R. And then last column here. Let me just check these. Okay, now let's go through this and let's count up how many. I mean, I've already got the results here, but let's confirm how many yellow, how many rounds, how many green, how many wrinkled. So, first of all, this is big Y will indicate yellow, big R indicates round, so that's yellow round, that's 1. That's yellow round. Oops. This is yellow round. Anytime you have a big Y and a big R, it's yellow round. And that's yellow round. Big Y, big R, yellow round. Just looking for yellow round. Big Y, big R, yellow round. Yellow round. Yellow round. And yellow round. I don't see any more. Should have a total of 9. 1, 2, 3, 4, 6, 7, 8, 9. So that's right. 9 yellow round, indicated by a big Y, big R. Okay, next, what about yellow wrinkled? Yellow would be big Y, wrinkle would be 2 little R's. So that would be yellow wrinkled. This would be yellow wrinkled. And this here would be yellow wrinkled. I don't see any more yellow wrinkled. 3, as we expected. What about green round? So green would be little y, 1 big R. So let's do that. So we have, oops, one here, green round, one here, one sec, the pen's gone on me. That would be green round, and then here, green round. So how many of them? 3 is expected. And then last but not least would be this one here. All lowercase, little y, which means green, and little r, which means wrinkled. So only one of those is expected. Okay, let's go on here just to fill out this back page. Turn to the back page where it says incomplete dominance. So what is incomplete dominance? Before Mendel, it was believed that parental traits were blended. In offspring and could not be separated in later generations. It turns out that they can be separated. Mendel's observations displayed simple dominance. With incomplete dominance, 2 phenotypes are blended. So the appearance is blended. But it can be separated. We'll get to that later. So it says here, with incomplete dominance, 2 phenotypes are blended rather than showing 2 different possibilities. So that's what incomplete dominance is. It's an apparent blending. Physical characteristics are blended. So, for example, when you have, this is a little unclear here, but this would be, we look at the white alleles here, that's little r, just for clarity's sake. And of course, that represents a white flower, in this case snapdragons, and big R represents a red flower, which is, it's got dominant, 2 dominant alleles. You can also have red if it's a big R, little r. So you think. Actually, we'll see in a second here, they blend. You have a big R, little r combination of alleles. It won't, in this particular case, end up looking red. It'll end up looking pink. And that's what we label incomplete dominance. Okay, so what do we have here? When we mix the 2 sets of alleles, we end up with, using a Punnett square, we get the following. We get all of this. 4 heterozygous combinations, big R, little r, which in the case of incomplete dominance leads to pink snapdragons, okay, which can be separated later on. You can separate these alleles by combining, in this case, these two here, snapdragons, and what would we get? Again, using a Punnett square, we'd end up getting this. We'd get back to the white, and then 2 pink and one red snapdragon. So we can separate the alleles in the F2 generation. Okay. You can also get similar separation when we combine the F1 generation with one of the parents, in this case the white. And again, using a Punnett square, we'd get half of them being pink and half the snapdragons being white. Proving, at least in this case, combining the different alleles, the big R, little r, results in what we call incomplete dominance, where there's a blending of colors. Okay, what is codominance? Codominance here. Codominance is where both of the alleles are expressed at the same time. So it's not a blending, but you see both separate types, in this case, colors. So both alleles are expressed at the same time. Okay, so here you go. This is an example here of codominance, both colors being expressed. This here is an example on the right of what we just covered, called incomplete dominance, where there's a blending of the colors. So codominance, both colors coexist at the same time. You see both of them. Incomplete implies blending. I'm going to end the lesson here, and then we'll go on to the next 2 or 3 pages in a separate video.